Hello everyone. In the previous video, we had learned about the fungi, its habitat and thallus organization. In this video, let us learn about the structure of a fungal cell. The fungal cell is eukaryotic in nature, which means the fungal cell contains a true nucleus. It has all the membrane-bound organelles which is present in a cytoplasm. The cytoplasm is enclosed by a cell membrane and the cell membrane is surrounded by cell wall. The fungal cell could be unicellular or multicellular in nature. The multicellular hyphae occurs as a filament. You can see in this image that this is one cell and this is another cell. The two cells are divided by a cross wall which is known as a septum. Now the septum is the continuation of the cell wall and the cell membrane. The orange line that you are seeing here is representing the plasma membrane of a fungi. Let us look into the cell wall of fungi. The cell wall of fungi is made up of chitin. The chitin is elongated in nature and they are fibular in nature, which are present on the cell membrane. Chitin is made up of a complex polysaccharide which is having a nitrogen content in it. Example of such polysaccharides are N-acetyl D-glucosamine. So chitins are made up of N-acetyl D-glucosamine. Apart from chitin, we also get to see cellulose, mannose and other glucose molecules in the cell wall of fungi. The manoproteins is known as proteins which are associated with mannose sugar. Mannose sugar along with proteins, we call them as manoproteins. Many glucose units together is called as polymer of glucose, which we call them as beta glucans. So the cell wall of fungi contains chitin, cellulose, manoproteins and beta glucans in them. Chitin gives the structural rigidity to the cell wall of fungi. Moving on into the cell membrane of fungi, the cell membrane of fungi or the plasma membrane of fungi is made up of phospholipid bilayer, which is clearly explained by a model known as fluid mosaic model given by Sanger and Nicholson. The phospholipid bilayer is having a phosphate head and it has a lipid tail. So the phosphate heads are hydrophilic in nature, so they are exposed outside, and the lipid layer is hydrophobic in nature, so they are embedded inside. Along with this phospholipid bilayer, in the cell membrane we also get to see certain proteins and sterols. These proteins could be integrated proteins. The proteins that are embedded in between the phospholipid layer is called as integrated proteins. Some proteins are present on the surface of the cell membrane. We call them as surface proteins. Along with this, we also have certain sterols present in the cell membrane. The sterols that is present in the cell membrane of fungi is called as ergosterol. The cell membrane of fungi is differentially permeable, which means it will allow only certain molecules to enter inside the cell. It will not allow all the components to enter inside the cell. When you look at the cell membrane of fungi under electron microscope, it appears as a tripartite structure. That is, there is an electron dense layer on either side of the cell membrane and the lesser dense region in the central region. The central region of the membrane is less dense compared to the region that are present on either sides. The phosphate heads appear as a denser region and the lipid tails appear as a less denser region under the electron microscope, which we call them as a tripartite region. Moving on, let us look into the cytoplasm. The cytoplasm of fungi is colorless in nature. They are embedded or surrounded by a cell membrane. Inside the cytoplasm, you get to see all the membrane-bound organelles, which includes endoplasmic reticulum, mitochondria, ribosomes, Golgi apparatus, vacuoles, inclusion bodies, and so on. The inclusion bodies will have stored food in them, or they might have certain oil or pigments or secretory granules that are present inside them. The cytoplasm near the tip of the hyphae. That is, at the apical region of the hyphae, the cytoplasm is denser in nature and they have so many vesicles in them. We call this region as the AVC region. AVC stands for apical vesicular complex. Now, this region has a lot of nutrients present in them which helps in the growth of fungi. This region is also called as Swid's N corpor. Swid's N corpor. That is about the cytoplasm. Moving on into the different membrane bound organelles. First, it is endoplasmic reticulum. Endoplasmic reticulum is highly vesicular in nature. Usually it is very loose and more irregular than in cells of green plants. The endoplasmic reticulum is of two types even in the fungal cell. It is smooth endoplasmic reticulum and rough endoplasmic reticulum. On rough endoplasmic reticulum, ribosomes are present. The ribosomes that are present in the cell of fungi is a 80S ribosome because it's a eukaryotic cell. Therefore, the ribosome is 80S ribosome, which is composed of 60S larger subunit and 40S smaller subunit, which helps in the synthesis of proteins. Now, the ribosomes that are present on the endoplasmic reticulum synthesize proteins and they will transport the proteins inside the endoplasmic reticulum. So, inside the lumen of endoplasmic reticulum, the protein modification is going to take place. Post-translational modification of proteins takes place inside the lumen of the endoplasmic reticulum. Lumen means cytoplasm like liquid that is present inside the endoplasmic reticulum is called as lumen of endoplasmic reticulum. The next cell membrane bound organelles or a cell bound organelle is called as mitochondria. Mitochondria is enveloped by a double membrane. The inner membrane is folded to form cristae. You can see this invaginations. The inner membrane of the mitochondria has invaginations. We call this invaginations as cristae. Now, as usual, the function of the mitochondria is to synthesize ATP. So, the ATP synthase enzyme is present on the inner membrane of the mitochondria, which helps in the synthesis of ATP. There is not much difference between the fungi of mitochondria and the mitochondria of green plants and animals. Moving on into the Golgi apparatus. The Golgi apparatus helps in the transportation of certain molecules. So they transport certain compounds from the cell to the outside of the cell, or sometimes it also helps in carrying certain compounds from the outside cell to the inside cell. So these vesicles that are present in the Golgi apparatus forms a transport vesicles which will carry certain compounds from and within the cell. Apart from these membrane bound organelles, we also get to see vacuoles, inclusion bodies and a true nucleus in the eukaryotic cell of a fungi. The vacuoles in the fungi, as they age, the vacuoles are going to enlarge and they are going to coalesce. Fungi cell also contains inclusion bodies. The cytoplasm has these inclusion bodies in the fungi. The inclusion bodies store food materials, lipid globules, protein materials and volutin granules. They also sometimes contain PHP granules which act as a stored food for the fungal cell, but they lack starch grains. Carotenoid pigments are also seen in inclusion bodies. Sometimes the inclusion bodies also contains chitin synthase enzyme. We call such inclusion bodies as chitosomes. So this chitosomes has chitin synthase enzyme in them which helps in the synthesis of chitin. Chitin is present in the cell wall of fungi. To synthesize the cell wall of fungi, chitin synthase enzyme is very essential, which are present inside the chitosomes. And then you have nucleus. Each cell of fungi contains nucleus which is globous or ellipsoid in structure. It has a central dense body. The nucleus has a central dense body with a clear area around it. The DNA is present as chromatin strands inside the nucleus, and nucleus is surrounded by a nuclear membrane which is having a nuclear pore in it. So the DNA is present in the form of chromatin strands. During somatic nuclear division, the cell undergoes mitosis. Mitosis means it has different stages like interphase, prophase, metaphase, anaphase, and telophase. The cell division is closed and thus termed as intracellular cell division. Now, let us look into the fungal flagella. The higher fungi like ascomycetes, basidiomycetes, and deuteromycetes lack motile cells in the life cycle of fungi, which means they will not reproduce any motile cells. The reproductive cells are non-motile in nature. In lower fungi, zygomycetes also lack motile cells, whereas some lower fungi which lives in the water like aquatic fungi has motile reproductive cells. Now these motile reproductive cells have flagella in them. Flagella are thin hair-like structures which emerges from the cell cytoplasm, whose function is to propel and helps the reproductive cells to move to new localities. The fungal cell is having a flagella. Now this flagella could be present in the anterior end or it could be present in the posterior end. Sometimes the reproductive cells will have only one flagella in them. Sometimes they might have 2 to 3 flagella in them. Let us look into the structure of a fungal flagellum. The fungal flagellum is made up of axonema. The core of the flagellum is called as axonema. Now this axonema is made up of microtubules. Microtubules are the cytoskeleton elements. Okay, so the microtubules is made the axonema is made up of microtubules. Now if you see, the microtubules are arranged in this fashion. The peripheral region of the axonema has 9 doublet microfibrillar structures, and in the central region there are 2 singlet microfibrillar regions. So we call it as a 9+2 arrangement. The axonema has 9+2 arrangement, or the flagellum has 9+2 arrangement of microtubules in them. Now these microtubules are surrounded by a cytoplasmic sheath. It has a double cytoplasmic sheath around it. So these microtubules are the ones which helps in the movement of the reproductive cells. We are not going to look into the mechanism of the movement of flagellum in this video. Now let us see the different kinds of flagella that are present in the fungi. Usually we get to see the 3 kinds of flagella. The first type is the whiplash flagella with an end piece. Now this image which you can see is a whiplash flagella. Now this whiplash flagella has a central axonema which is surrounded by a cytoplasmic sheath, and at the end the axonema runs into to form an end piece. So whiplash flagella with an end piece is the first type of flagella that we are going to see. The second type of flagella is the whiplash flagella with a blunt end. You can see this is the whiplash flagella with a blunt end. Now here this flagella has a axonema and it is surrounded by a cytoplasmic sheath. The axonema will not form a narrow tip, so therefore it has a blunt end. And then the third type of flagellum is called as a tin cell flagella. Now this tin cell flagella is having so many hair-like appendages around the cytoplasmic sheath. Now this hair-like appendages arises from the axonema. So the tin cell flagella also has a axonema which is surrounded by the cytoplasmic sheath, and there are a lot of hair-like appendages present around this cytoplasmic sheath. Tin cell flagella is also called as pantonymatic flagella, flimmer or flimmer-giesel flagellum. The whiplash flagellum is also called as acronematic or pitch-giesel flagellum. The arrangement of flagellum is known as flagellation. Let us now look into the different types of flagellation usually that is seen in the reproductive cells of the fungi. The first type is a single whiplash flagella is seen at the posterior end of a reproductive cell. Now the back end is called as the posterior end and the front end is called as the anterior end. The first type is the single whiplash flagella at the posterior end, which is usually seen in the reproductive cells of the chytridiomycetes. You have another type of flagellum, that is the B type, where the single tensile flagella is found at the anterior end of a reproductive cell, at the front end of a reproductive cell. So it is usually seen in hypochytridiomycetes. And then you have a biflagellate. Biflagellate means two flagellas at the anterior end. The one flagella is shorter. The whiplash flagella with a blunt end is shorter in this type when compared to the whiplash flagella with a narrow tip. So it is having two flagella, both are whiplash, whereas one is with the narrow tip and the other one is with the blunt end. And the fourth type is also again a biflagellate reproductive cell, which is usually seen in the pyriform shape where it is present at the anterior end. Now here one of the flagella is a whiplash flagella and the other flagella is a tinsel flagella. And the fifth type of flagella which is seen is again a biflagellate reproductive cell, where one flagella is of tinsel type and the other flagella is of whiplash type, whereas here both these flagellas are going to arise in a kidney shaped cell where there is a depression in the kidney shaped cell. Near the depression, from there the flagella is going to arise. Now the tinsel flagella is present in the forward region and the whiplash flagella is present at the backward region. The whiplash flagella is so many times lengthier and longer than the tinsel flagella. It is usually seen in Oomycetes family. Now let us move into the modification of hyphae. The modification of hyphae is not a part of the structure of a fungal cell, but it is a very small topic and it is in an important topic. That's why I have included this in this video. So fungi sometimes will get modified into different shapes. Like many hyphas of fungi will come together, they aggregate to form tissues. So plectenchyma is a false tissue which is formed by the aggregation of the hyphae. Plectenchyma is a false tissue which is formed by the aggregation of hyphae. There are two types of plectenchyma. We call them as prosenchyma and pseudoparenchyma. Prosenchyma is made up of a loosely woven tissue of a hyphae. You can see many tissues many hyphae is going to aggregate to form a prosenchyma. Here the hyphae are elongated in nature and they are present parallel to each other. So here the hyphae is going to run parallel to each other to form prosenchyma and it is loosely arranged. Now this prosenchyma will not each hyphae in the prosenchyma is not going to lose their identity when they form a tissue, whereas the pseudoparenchyma is a complex hyphae, that is hyphal modification that is seen in fungi, where the hyphae is going to get compactly packed. Here the hyphae is tightly wound. If you see in prosenchyma it is loosely woven, whereas in pseudoparenchyma it is tightly woven. The walls of the hyphae in the mass gets fused and they lose their individuality in pseudoparenchyma. It appears as a isodiametric or oval cells. It resembles the parenchyma tissue of the higher plants, therefore it is called as pseudoparenchyma. Sometimes the hyphal cells also forms rhizomorphs. The hyphal cells will aggregate to form rhizomorphs. Now the rhizomorph is a thick strand or root-like aggregation of a somatic hyphae. it loses its identity because it gets fused. The hyphae gets fused and it loses its identity. The whole mass here is going to behave as an organized unit and a single unit. Rhizomorphs helps in the absorption of nutrients from the substratum. The substratum is the body on which the fungi grows. If the fungi is growing in a soil, then soil acts as a substratum. So the rhizomorphs, the aggregation of fungi which forms a rhizomorphs, helps in the absorption of nutrients from the soil or from a substratum. The fungal hyphae also forms sclerotium. A sclerotium is a compact globus or a elongated structure formed by the aggregation and adhesion of the hyphae. The sclerotium is the resting phase of hyphae. During reproduction also you get to see sclerotium. Sclerotia is usually seen in Claviceps purpurea, where the fungal hyphae will aggregate to form the sclerotia. During unfavorable conditions, the fungal hyphae will aggregate to form sclerotia, and it acts as the resting phase of fungi. It acts like a spore. Whenever there is favorable conditions, the sclerotium will germinate to form a new individual. The other type of hyphal modification that you get to see is a stromata. A fungal tissue that forms a reproductive structures are called as stromata. Now the stromata is usually seen in Basidiomycetes family. The stromata, if I have to tell in simpler words, the stalk of the mushroom is a false tissue which we call them as stromata. So the stalk of the mushroom is actually a stromata. The stromata will bear reproductive cells. For example, the reproductive spores are present inside the fruiting body of a mushroom. So the mushroom stalk is the stromata. Apart from these modifications, we also get to see the hyphae gets modified into two other structures, that is aprosorium and hostorium, which helps in the drawing of nutrients from the host or a substratum. Certain fungi are parasitic in nature, which means they absorb nutrients from a living host. So the parasitic fungi will form two types of structures. The hyphae of a parasitic fungi will form two types of structures. We call them as aprosorium and hostorium. Now this aprosorium is the singular form and aprosoria is the plural form of aprosorium. They are usually found in the parasitic fungi, mostly the ectoparasites. When I say ectoparasites, outside, the parasites which stay outside the host cell, we call them as ectoparasites. And aprosorium is a terminal, simple, lobed swollen structure. You can see it is a lobed swollen structure which is arising from a spore. The spore produces a germination tube. Through the germination tube arises a hyphae. Now this hyphae is going to get swollen and they form the aprosorium. Aprosorium helps in the adhesion of the fungal hyphae onto the surface of the host. Once the fungal cell adheres onto the surface of the host, the hyphae will get inside the host cell and they will start drawing nutrition from the host cell, the living host cell. The host could be a plant or an animal. Now this hyphae which enters into the cell is going to form another structure. We call them as hostorium. Hostorium is the singular form and hostoria is the plural form. They are produced, this hostorium are produced as intracellular absorbing structures in an obligate parasites. These hostorium vary in shape. It could be globus in shape. You can see they could be round in shape or sometimes they could be branched. Now this hostorium is the one which is going to enter into the host cell and absorb the nutrients from the host without killing the host. The ostorium is going to increase the surface area of a hyphae so that it can penetrate inside the cytoplasm and absorb all the required nutrients from the host cell. More about Hostorium and Aprosorium we will learn in the nutrient part of fungi. Whenever we are learning the nutrient requirements in fungi or a mode of nutrition in that unit, we are going to learn more about this Aprosorium and Hostorium. Here the hyphae here in Aprosorium is getting modified, it is getting swollen, which helps in the adhesion of parasitic hyphae onto the host surface, and Hostorium will enter into the host cell and they will absorb nutrients from the host cell. The Hostorium could be round in shaped, that means it is a knob shaped or a branched finger shaped. You can see this is the branch finger shape. So these are the different hyphal modifications that we usually see in fungi. Thanks to all of you for listening to my video, and in the next video let us learn about the nutrition in fungi and the different modes of nutrition that the fungi undergoes. Thank you all.